Hello, my name is Michael Reidenbaugh, and today I'm going to be presenting on research that I performed in my microbiology course. The goal of this research was to find bacteria in soil that is able to produce undiscovered antibiotics. This is important because antibiotic resistance, if you haven't heard of this, is whenever bacteria are becoming immune to the antibiotic drugs that we use to treat them. And this mainly happens due to overuse and misuse of antibiotic drugs. And since bacteria just keep getting more and more resistant to the drugs that we use now, the best solution is to just discover new drugs that bacteria do not resist. And so the soil that I used was from a forest in Washington County of 84, Pennsylvania. And the 1 gram of this soil was put in 10 milliliters of water, and this was serial diluted several times to make spread plates. And these spread plates were used to find isolated colonies of bacteria. 12 isolated colonies were used to form a master plate, which is in figure 1. This is basically a reservoir for all of the isolates, so that if I need to come back and get a new sample, they're just right there. And all 12 isolates grew. A few were similar in their characteristics, but there was a fair amount of differences. The morphologies of each of the isolates were described based on their margins, colors, elevations, shapes, and textures. And as I said, there's a few similarities between some of the isolates. So what I did next is I created bacterial lawns of Escherichia coli, Staphylococcus epidermidis, and Pseudomonas aeruginosa. And then I placed 12 of the isolates onto each of these lawns to test for antibiotic production. And isolates 4 and 7 actually were found to be antibiotic producing, and they only were seen to affect Pseudomonas aeruginosa. As you can see within the blue circles, there is a small area of no growth around the colonies. These are called zones of inhibition. And these were fairly large by comparison to what you could have seen. So they are noticeable. So what I did next is I attempted to describe both of the antibiotic producing isolates. So isolates 4 and 7 underwent gram staining and a few biochemical tests to kind of see what they are. For the gram stain, both were seen to be gram-negative bacilli, so small pink rod-shaped bacteria. There were oxidase and catalase tests. They were the same for each of those as well. For oxidase testing, both were negative, and for the catalase test, both were positive. And next, eosin methylene blue agar, both isolates were plated on that. And as you can see in figure 5, there is pink growth. This again confirms that they are gram-negative. However, since they're pink instead of a dark black purplish color, this shows that they are non-lactose fermenting. And so when performing these tests, I was first contemplating that these were the same organism, since they looked the same and they had a lot of the same biochemical properties. However, the gelatinase testing really showed me wrong. As you can see in figure 6, isolate 7, which is on the right, was able to break down the gelatin into a liquid, while isolate 4, which is on the left, could not. And this was the most significant test, since it showed that both of these were different organisms and not the same. So the overall results are quite significant, because not 1, but 2 bacterial species were discovered to be effective against Pseudomonas aeruginosa, which is a fairly dangerous bacterial species. And so in the future, what can be done is we can test the antibiotic compound against even more bacterial species to see if it's effective against them. I only tested against 3, but there's a lot more infectious bacteria out there. What can also be done is to attempt to find the identity of these isolates and see if they're known species that are already known to produce antibiotics. But if they're not, if these are seen to be new species, or if we haven't known these species to produce antibiotics, what can be done is we can attempt to describe the antibiotic compounds themselves and see how they specifically interact with the bacteria that they fight against. And if the drug looks good, what is the overall goal is to make an antibiotic drug that is FDA approved. Overall, this study was a success, and it showed that antibiotic compounds can be uncovered from soil samples. And it's quite significant that we can find just antibiotics from soil, since resistance is becoming a big problem, and students across the world are just able to discover new antibiotics. It's a numbers game. If everyone begins to look for antibiotics, then we may just find them, and we would be able to treat a lot of diseases quite easily. Thank you for listening.